On His Majesty's service. Synapses. Synapses are structures that transmit nerve impulses from one neuron to another, the so-called interneuronal synapses, or to effector cells, that is, the neuroeffector junctions. In this video, interneuronal synapses will be described. They are classified as electrical and chemical. The electrical synapses are common in invertebrates. In mammals, they are found in some nuclei of the thalamus, neocortex, and hippocampus, and are represented by gap junctions. Chemical synapses transmit impulses from a presynaptic neuron to another cell using specific biologically active substances, neurotransmitters. Morphological classification of synapses takes into account which parts of the contacting neurons are involved in the formation of a synapse. According to this classification, there are exadendritic, exasomatic, and exaxonic synapses. In some parts of CNS, dendrodendritic, dendrosomatic, or somatosomatic synapses could be found. In exadendritic synapses, axon usually ends on the dendritic spine, more rarely on the dendritic shaft. The chemical synapse consists of three parts: presynaptic region, or presynaptic knob, or synaptic bouton. It is the expanded end of the axon. Synaptic cleft is the 20 to 30 nanometers gap between the presynaptic and postsynaptic membranes. And postsynaptic region, it is the area of the membrane of the postsynaptic neuron where receptors with which neurotransmitters interact are localized. The presynaptic region includes synaptic vesicles with neurotransmitters, mitochondria, smooth endoplasmic reticulum, and presynaptic plasma membrane. Voltage-gated calcium channels are localized on the presynaptic membrane. Cytoplasm near the presynaptic membrane is rich in clusters of biologically active macromolecules and proteins. It is called presynaptic density and possesses active zones where some synaptic vesicles are docked, that is, are in contact with the plasma membrane. They are primed, that is, ready to fuse with the membrane upon calcium ions elevation. These synaptic vesicles constitute the so-called readily releasable pool that is immediately available for fusion upon the arrival of an action potential. Receptors for neurotransmitters are localized on the postsynaptic membrane. Cytoplasm near the postsynaptic membrane also contains accumulations of proteins and is termed postsynaptic density. When a nerve impulse, that is, the wave of the membrane depolarization, reaches the synapse, the voltage-gated calcium channels open up, which leads to the influx of calcium ions from the extracellular space to the cytoplasm. The synaptic vesicles of readily releasable pool fuse with the plasma membrane and release neurotransmitter into the synaptic cleft by exocytosis. the neurotransmitter binds to receptors that may be ionotropic or metabotropic. The ionotropic receptors possess ion channels. The binding of neurotransmitters to them results in conformational changes in the channel's proteins that cause their pores to open. The response depends on the identity of the ions that enter the cell. For example, the influx of sodium ions causes local depolarization of the postsynaptic membrane and can generate nerve impulse in the postsynaptic neuron. Such synapse is called excitatory. The influx of chlorine ions leads to hyperpolarization of the postsynaptic membrane, making the generation of an action potential impossible. This is the inhibitory synapse. Some neurotransmitters bind to G-protein-coupled receptors, which are metabotropic. They activate G-proteins, which in turn activate effector proteins, G-protein-gated ion channels, or primary effector proteins. The latter then generate second messengers, which in turn activate one or more secondary effector proteins. The secondary effectors initiate a wide range of intracellular signaling cascades that affect many aspects of cellular function and signaling. Thus, synapses may be excitatory or inhibitory. Most neurons in CNS receive inputs from thousands of synapses. Some of them are excitatory, some are inhibitory. Spatial and temporal summation of excitation and inhibition by postsynaptic neuron results in the integration of the electrical information provided by all the synapses acting on it at any moment. The production of an action potential depends on the balance between excitation and inhibition. After a neurotransmitter molecule binds to a receptor and activates it, the neurotransmitter is removed to end the signal and clear the way for the next signal. It is done through one of such processes. The neurotransmitter may diffuse away, making it available to be broken down metabolically outside the neuron, for example, by astrocyte. Pumps within the presynaptic membrane may actively pump the neurotransmitter back into the synaptic bouton for reprocessing and re-release. Enzymes within the postsynaptic membrane may inactivate or metabolize the neurotransmitter. To support high rates of transmitter release, synapses require a constant supply of neurotransmitter-filled synaptic vesicles. Synthesis of synaptic vesicles in the pericardia and axonal transport to synapses is too slow. The number of synaptic vesicles that undergo exocytosis over a given time period can greatly exceed the supply of synaptic vesicle precursors delivered from the cell body. Thus, this supply is maintained by either rapid reuse of fused vesicles or requirement of new vesicles from the so-called reserve pool. A readily releasable pool and a fraction of the reserve pool that participate in activity-induced synaptic transmission are termed the recycling pool, and the process of reconstituting synaptic vesicles is termed synaptic vesicle recycling. The remaining synaptic vesicles are called the resting pool, which can be mobilized during or after very prolonged stimulation. After neurotransmitter release, the membrane of the fused vesicle diffuses laterally to the area outside the active zone, where it is retrieved by clathrin-mediated endocytosis. Then they are refilled with molecules of neurotransmitter. Recently, several clathrin-independent mechanisms of endocytosis have been identified. They are widespread in cold-blooded animals, but also were shown in mammalian CNS, and it is possible that all mechanisms coexist in synapses and are used differently depending on activity levels or synapse type. And it is important to note that many specific proteins are involved in providing and control of synaptic vesicle recycling. The mode of exocytosis described above is called full collapse fusion. In another mode, a pore connecting the lumen of the vesicle with a synaptic cleft is formed without the vesicle collapsing fully into the plasma membrane. Neurotransmitter can then be released in a controlled fashion through this transient pore by porocytosis. Thus, each synaptic vesicle can be used multiple times to release neurotransmitter during burst in neuron activity. This mode of exocytosis is called kiss and drum fusion. The role of neurotransmitters can be performed by various substances. The total number of neurotransmitters is well over 100. The common are acetylcholine, nitric oxide, ATP, amino acids, glycine, glutamate, gamma-aminobutyrate, aspartate, some biogenic amines, norepinephrine, epinephrine, serotonin, dopamine, and peptides, neurotensin, vasopressin, somatostatin, etc. Neurotransmitters, except nitric oxide, are stored in synaptic vesicles. After the rise in the concentration of calcium ions, nitric oxide is synthesized by NO synthase within the synapse boutron and immediately diffuses to the surrounding neurons, gliocytes, and other cells in the vicinity of a few tens micrometers and produces a wide variety of biological effects. It was believed earlier that every neuron produces only a single type of neurotransmitter. Now it is clear that many types of neurons synthesize and release two or more different neurotransmitters, which are called co-transmitters. They are packaged in different populations of synaptic vesicles and need not be released simultaneously. Co-transmitters are differentially released according to the pattern of synaptic activity. Low-frequency stimulation releases one type of neurotransmitters. High-frequency activity releases another type or both neurotransmitters. Rod and cone cells and bipolar neurons in the retina and hair cells in the inner ear have specialized ribbon synapses. They possess an electron-dense proteinaceous organelle, the synaptic ribbon, which is anchored to the cytomatrix at the active zone of the plasma membrane density. About 100 or more synaptic vesicles containing glutamate are tethered on the synaptic ribbon. They provide a pool for sustained release that is 5-fold greater than the docked pool available for release in conventional synapses. release of synaptic vesicles occurs in response to calcium ions influx through voltage-gated calcium channels, which are located beneath the ribbon. The quantity of fused synaptic vesicles depends on variations in the membrane potential of the receptor cell. Thus, ribbon synapses provide continuous and high-level exocytosis controlled by graded membrane potential changes. Retrieval for vesicles occurs mainly through clathrin-mediated endocytosis. Thank you for your attention.